Hi everyone, welcome to Maths World UK. I'm James Grine and today I'm speaking with Jen Rogers. Jen is a statistician and Vice President of PHASTAR, which is a consultancy in analysing clinical trials. So they've been very busy this past year and they've been designing and analysing the vaccine trials for COVID. And so Jen's agreed to answer some frequently asked questions about the vaccine trials. For example, how they collect good data about COVID so we know how fast it spreads, and also how we got the vaccine so quickly while still being safe. But first, I started by asking Jen about some of the basics of a clinical trial.
So individuals get randomized. So first of all, you'd need to decide which treatment a person gets. So typically when you do these clinical trials, you compare your new treatment to either a standard treatment that already exists or a placebo if there isn't anything that already exists. And what you would do is you would give some people in your study your new treatment and you would give some people your standard or placebo treatment. And first of all, you need to randomize the individuals. So we don't just, say, take the top half and give them one and then the next half and give them the other. We have to formally randomize which group people go in. Then you follow them up through time and you have a look. It might be that, you're wanting to, prevent headaches or something like that. And you would count how many headaches there were in each group. And you would see if there were then differences between the two groups. And that would hopefully tell you whether or not your new treatment or device worked.
So the idea is hopefully at the end, the new drug that you're testing means they have less headaches than the group that had the placebo.
Yeah, exactly. And, there's lots of formal statistical analyses that we can do to say that whether or not it works. Then we would sort of package all that up and send it off to the regulators.
And this is all very relevant now because everyone's suddenly very interested in how clinical trials work.
Yeah, I never thought I'd overhear, conversations that people would be having talking about statistics. And I think right now the major questions that I get asked are around the vaccine. Is it safe for people to take? That's the big question. And I get that, we're all putting a new medicine into our bodies, or we're all being asked to put a new medicine into our bodies. And the speed at which these vaccines have been licensed, I think, is what makes some people a little bit nervous and worried about whether or not corners have been cut and whether or not they are, should they be taking it?
Well, so you've kind of sort of prepared some of the questions that you do get asked a lot. So maybe I'll just hand it over to you.
So throughout the coronavirus pandemic, there's been an absolute flood of data, and we need to try and wade our way through all of that to answer lots of questions, such as how does COVID-19 spread? Where is it now? How many people have got it? And what treatments are safe and effective? But the difficulty with this pandemic is that it's a brand new disease, and everything's changing all of the time. We're in lockdown, we're not in lockdown. We're changing the number of people we're testing. We've now got vaccines. The environment's been changing all of the time. And so trying to make sense of all of this data in this fast changing environment is a really difficult task. Even the most simple question, such as how many people have got COVID-19, isn't easy to answer. We would hope that reported daily number of cases that we see, the government releases those every day, we would hope that would be a pretty good proxy or estimate for how many people might have COVID right now. But unfortunately, that's not necessarily the case. Those numbers that we see don't necessarily give us a good estimate of what the overall prevalence is in the UK. Now, one of the most simple reasons why that might not be the case is we've been testing differently all the way throughout. The number of people who have been testing and who we've been testing has been changing all the way through this pandemic. And that then has a really big effect on the relationship between that estimated daily reported number of cases, and the estimated prevalence in the population. Now, we have studies such as the Office for National Statistics Infection Survey and a REACT study that's been run by mainly Imperial College London. And they give us much better estimates as to how many people have coronavirus in the population. But the problem with that is that this data is slower to collect. We typically see this usually about 2 to 3 weeks lag at any one time. It's really important that the government are able to make quick decisions based on what's going on right now. And so really there's this weighing up between data that is fast, but might be quite messy, and better data that is slow. And it's a really difficult balancing act between trying to make decisions based on this sort of messy data versus slower, better data. Even if we look at death data, you'd think, a death is a death. That should be easier to try and analyze and, report and monitor, but that's also not without its issues. One of the problems associated with the death data that we see from the government every day is the way that they define a COVID death. So they define a COVID death as a person who has had a positive test result for COVID-19 and then died within 28 days of that first positive test. So that means that if a person had a positive COVID test on one day and then the next day was knocked down by a bus and killed, they would be classed as a COVID death. Alternatively, if a person received a positive test for COVID and then they died on day 29 from COVID, they wouldn't be classed as a COVID death. So there's these issues with how they define what this COVID death is. But again, it's this data that's quick and it's easier to collect, and we can see it on a daily basis, which allows us to make decisions quicker. A more reliable way to look at COVID deaths is using, again, the Office for National Statistics, ONS. They actually look at people's death certificates. So they see whether or not COVID was mentioned on their death certificate as a cause of death. And that's how they define their COVID deaths. So again, we see that the data that we're getting from somewhere like ONS, it's better quality data, it's more reliable data. But again, this data is slower to collect. And we have, again, this lag of about 2 weeks between the person dying and then appearing in the reported case numbers from ONS. And so, again, it's this really difficult balancing act between messy, quick data and slow, reliable data. And that's really been a theme throughout all of this. And it doesn't mean that none of this data should be used. They all have different purposes, but I think what's really important is that we need to make decisions quickly, but we need to be open to changing those decisions as new data comes to light. Now, I just want to finish by talking about vaccines very quickly. So we've talked about cases, we've talked about deaths, and I just want to talk about vaccines because I think when will we get a vaccine was probably one of the biggest questions that was asked in 2020. It was a huge topic of conversation, and I think the whole world probably breathed a huge sigh of relief when we got that first press release on the 9th of November from Pfizer-BioNTech saying that they had a vaccine that worked. It seemed like that there was potentially now some light at the end of that very long tunnel. But there are lots of questions that people do ask. It is a massive achievement that we've been able to put these vaccines through all of the processes so quickly and license them in record speed. But it does mean that there is some uncertainty from the general public about whether or not this vaccine works and whether or not it's safe. So first of all, I just want to say, okay, we are told that, for example, the Pfizer-BioNTech vaccine has an efficacy or an effectiveness of 95%. And the first question is, what does that actually mean? What is a vaccine effectiveness of 95%? It doesn't mean that you've got a 5% chance of getting COVID after you've had the vaccine. I think that's what most people think. And that's what people tend to say to me, oh, that means I've only got a 5% chance of getting COVID after the vaccine. It's not quite what it means. It's close, but it's not entirely right. What a vaccine effectiveness of 95% means, it means if you were to take 100 people who definitely would get COVID, if those then 100 people were vaccinated, you would only expect 5 of them to then get COVID. So it's slightly different from having a probability of 5%. It actually means your probability of getting COVID is even smaller than that, because not everybody has a 100% chance of getting COVID. It just means if you were to get COVID, now you've only got a 5% chance of getting COVID. The next question that I think I get asked is, does the fact that it's been approved so quickly mean that shortcuts have been taken and corners have been cut? And, does that mean then that it's unsafe and I should be worried? And I think I would just say very simply, absolutely not. So usually when vaccines get developed, we talk about clinical trials. There are lots of different phases of clinical trials. So we have phase 1 through to phase 3. And typically all of those phases would be run one after another. So you design your phase 1, then you do it, then you'd analyze it, then you design your phase 2, you'd do it, you'd analyze it, and so on and so on. And at every single point, you'd be talking to regulators. And all of that takes a long time. You might need to get funding for it and things like that. But in this instance, they haven't been running them sequentially. So normally you'd run them sequentially because it wouldn't make sense to run your phase 2 until you knew that your phase 1 worked. It wouldn't normally make sense to start manufacturing a new drug until you knew it worked. But because every single day has meant people have been dying, people aren't so worried about making a vaccine that then turns out not to work and wasting money. They'd rather just speed up the process as quickly as possible. And so all of these different phases that would usually run one after another have been run sort of alongside each other. And that's perfectly fine to do. And whilst these trials have been sort of squished or sped up in calendar time, the person time spent in these studies hasn't been any different from any other sort of clinical trials that you might have for any other treatment. So yes, they've been done quickly, but no corners have been cut, and everything that should have happened has happened. We have tested it on enough people to be sure that it works. There's some people are saying, there were only sort of 170 cases. Is that enough evidence to say whether or not it works? When we carry out studies, we do formal sample size calculations. And for example, the Pfizer-BioNTech study, they calculated that they would need to see a vaccine effectiveness of at least 50% in 164 cases to be able to have enough evidence to say their vaccine worked. What they actually saw was a 95% effectiveness in 170 cases. So they definitely did have enough evidence to say that it worked. So yes, these studies have been done really quickly. Unfortunately, there's no such thing as zero risk ever. Even when you take a paracetamol, there is a risk associated. But weighing up the risks alongside the benefits, we can be sure that these vaccines, they are, everything indicates that they are safe, they are effective. And my 3 grandparents have now all been vaccinated. They've taken their first vaccine, and I was super happy. So yeah, it's been a very challenging year for medical statistics. It's great to see data being discussed and the reliability of data and intricacies of data, taking 7-day averages versus looking at it on a granular level. I think things like that are just great to be seen, being discussed in a public arena. But yeah, it's difficult, it's challenging. And I've written loads of different blogs on all of these in more detail, which, I'm sure you could look up if you wanted. But yeah, there you go. The challenges of being a medical statistician during a global pandemic.
I think those are the important questions that people will be thinking of, want to answered. Can I ask you a couple of other questions?
Yeah, sure.
I don't know if these are good questions or not, but we've got 3 vaccines at the moment, I believe. Is that correct?
Yes.
That's Pfizer,
yep.
Moderna,
yep.
and the Oxford.
Yep. So yeah, we've got Pfizer and BioNTech, Moderna, and the Oxford AstraZeneca. Yep.
And can we compare these vaccines if they've got different trials? So maybe they've been people have been given a different placebo, they've been run differently. Can we compare these vaccines?
Yeah, we've seen lots of talk about which vaccine is best and kind of wanting to create this hierarchy of vaccine effectiveness. You're right, you can't really directly compare the results from the different studies. The way that they've defined a COVID case is different in some of the studies. So I think it's Moderna, maybe, they defined it as from 2 weeks after their first vaccine, whereas Pfizer only considered cases that were 1 week after the second vaccine, or something like that. That might not be, verbatim true, but the fact is that the way that they defined a COVID case was slightly different for the different studies. Pfizer and BioNTech and the Moderna study, they used a placebo control, whereas in the Oxford AstraZeneca study, in the UK part of it anyway, they used what we call an active control. So it wasn't a placebo, it was actually the meningitis jab. And the reason that they did that was because if you had a placebo, you might get no side effects. So like no headache, feeling a bit unwell for a couple of days, which you typically tend to get with vaccines. Whereas if you give someone a meningitis vaccine, they may have those same side effects, and it just helps with the blinding. Ideally, you don't want people to know which treatment they've been allocated to. And if you get side effects, you might think, oh, well, I've obviously had the vaccine then. And if you get no side effects, you might think, oh, well, I've obviously been given the placebo then. If you give someone an active control, you avoid that happening.
So we've got a new variant now, which is even more transmissible. Yeah. Does it affect the speed of your trials? I mean, can you get results faster? Can you learn more if it is spreading faster?
Yeah, I mean, it's really horrible when you work in clinical trials that you need events to be able to say whether or not treatments work. To be able to see whether or not there's a difference between two treatment groups, we need to see how many cases there are. And so studies have actually been really struggling when we've not had as many cases. And so we've been having to sort of recruit more people into studies and things like that. So once the prevalence in the population goes down, you need more and more people because you need to see those cases. And so, yeah, having a new variant that spreads quicker means that we see more cases quicker, and it means that we'll be able to see differences between the two treatment groups quicker. So you find yourself sometimes in a horrible situation where you're saying, I need more events, I need more cases. And that's so counterintuitive to what we're trying to do in the general population, which is bring case numbers down.
And so we're now at the stage of rolling this out to the entire population.
Yeah.
So what happens next in your analysis? So you're going to have millions of points of data.
Yeah, so I think some people think that when we've done our clinical trial and it gets approved by the regulators, that's sort of it, and it's free to roam into the general public, and that's not the case. We do follow on studies. You want to be certain that the vaccine effectiveness that you saw in your phase 3 study actually does carry on throughout the general population. And you want to be making sure that, safety data is still okay, because, some safety events might have a 1 in 10 million probability. And no matter how big your study is, you're never going to see that. So we have to keep monitoring everybody. So we have what are called phase 4 studies, which are surveillance and confirmatory studies. And those will be carried on. So people will be followed up, people will be monitored to see, is the effectiveness still what we thought it was? Is it safe? People will be able to submit any side effects that they get as well. So the MHRA has what is called a yellow card system. So you can go online, and if you've had a side effect from any medication, anything at all, even if a paracetamol you had a side effect, you could go on there and you could log it. And then they just get monitored, and there's all sorts of AI being brought in to sift through all of this to pick out, okay, well, these are just headaches, these are just mild, versus these are the really serious ones that we need to be looking at. So it's not the end of the story by any stretch of the imagination, and all of these studies will be carried on for probably many years.
So thanks for coming and answering our questions. Is there anything that you want to plug before we go?
Well, if you go on my company, so PHASTAR.com website, there is a blog, and I've written lots of different articles. So challenges of looking at the case data, challenges of looking at the death data. And I did a write-up every time one of the vaccines was approved. You can get them all on my website as well. I've got a little COVID section. It's a joyful read where you can see all of the different things. And yeah, you can read up and see what's safe and how effective they are and what are the challenges of this messy data in more detail.
Hi, thanks to Jen for coming in and speaking to us. If you want to find out more, you can find the link to Jen's blog in the description. And that's all from me. So I'll say stay safe, stay curious, and I'll see you next time.